section, we will try to understand that how is information in DNA used to make the proteins that control cells' activities. Again, this process is transcription and translation. Some of you have probably most likely covered it in other biology classes, so this should be a good review. In the process of transcription, genetic information is copied in DNA into a sequence called as mRNA, and in translation, that mRNA is later processed through the process of translation to synthesize a protein. So let's look at how does DNA direct protein synthesis. To understand that, we need to first review the different kinds of RNA. The DNA will not directly form the protein without the help of the ribonucleic acid. The ribonucleic acid are of three different kinds. T stands for transfer RNA, R stands for ribosomal RNA, and M stands for messenger RNA. So there are three kinds of RNA that are involved in this process. The first one we want to look at is the mRNA. mRNA stands for messenger RNA, and it carries the coded information from making specific proteins from DNA to the ribosomes where the proteins will be synthesized. The formation of mRNA from DNA is called as transcription. So in transcription, pretty much what happens is that a strand of mRNA is synthesized using the specific regions of the DNA and forming a single strand of ribonucleic acid. For example, on this picture over here, you can see that the purple ones, A, C, T, G, C, these are all the strands on the DNA. And the RNA is formed, which is complementary of adenine. So RNA does not have the nucleotide thymine. It has uracil at its place. So uracil is forming. The next one is it's reading cytosine. So the complementary nucleotide would be guanine. Following that, it's reading thymine on the DNA. So it will give it will give A, or it will get RNA. Adenine nucleotide, and so forth. So pretty much what it does, it copies the DNA to create a single strand of a nucleotide. Now this particular strand is now called as the mRNA. mRNA then will leave then leave that region of the DNA in a prokaryote, and in a eukaryote it will leave the nucleus, and it will go towards the ribosomes. Ribosomes are regions that read the genetic code in triplets. So all these nucleotides that are coming from the mRNA will be read in a pair of 3 to then deliver a proper amino acid. So, and I'll show you later what I mean by this. So let's again go back into transcription, and the mRNA that is created often in translation, what I'm trying to say is often translation will occur while transcription happens. So that means translation cannot take place without transcription. So transcription has to take place, because in transcription, the original DNA strand is transcribed, which is pretty much copied down as a single strand. And in doing that, the thymine nucleotide is replaced by uracil. The process begins with the enzyme RNA polymerase, which reads a region on the DNA, and this region on the DNA where it reads is called as a promoter region. So promoter region is actually a region that will signal the RNA polymerase to bind at a particular site. Only one of the two strands serves as the template for RNA synthesis for a given DNA strand. And RNA is also synthesized from 5' to 3' direction. Now, in this particular image, we've actually shown you, and this is a picture from eukaryote because you can see a nucleus and a cytoplasm, but a lot of the steps are the same in the prokaryote, so I just like this image I wanted to share over here. Now, notice on this picture, what is something shown is the region called exons and introns. Introns are regions that that does not have any code. So introns are actually sliced out of the RNA, and the RNA which is mature or processed has a shorter base length. So notice all the introns are gone, so you have the shorter mature RNA, which will then go towards the ribosome for protein synthesis. Now, what happens in translation is that ribosomes attach to the messenger RNA. Now, notice in a bacterial cell, transcription and translation are going on in the same chamber. So once transcription is done, it's going to be followed by translation. Now, when in translation, the mRNA is carrying any of these 64 combinations of code. Notice each of these codes are 3 letters, or they are triplets, and each of these are coding for a unique amino acid. So, for example, triple U over here, it's coding for phenylalanine, but also UUC is also coding for an amino acid called phenylalanin. UGU is a code for cysteine. UGC is also a code for cysteine. Now, we call this as redundant or degenerate, that there are more than same code for more than one code for a particular amino acid. Now, notice over here for leucine, we have 4 different codes. So the total combination is actually not 61, it's 64 combination. You may want to fix that. There are 3... well, the reason we had 61 was, okay, I got it. Why I put that is because we have 3 stop codons. So notice over here we have the stop codons, and then we have 1 start codon. The stop codons are actually nonsense codes, which has absolutely no sense. So when the mRNA is being read, and when the stop codon is read, that signals the ribosome to stop making that elongated polypeptide, and the protein is pretty much made. So start codon is always the same, and you can see from here it's always AUG. That's the start codon. And it will start in bacteria, it codes for formylmethionine, and for eukaryotes it codes for methionine. Now, tRNA brings an appropriate amino acid. AA stands for amino acid, which matches the particular anticodon. So let's talk about what is an anticodon. Now, once the mRNA is being read by the ribosome, notice the ribosome in this strand is showing you the mRNA, and it has 3 start codons. Now, this is read by this little piece over here, and this little piece is the transfer RNA. The transfer RNA brings an amino acid on its one end, and it has a triplet code on the other end. The triplet code codes, for example, UAC, and that will be the complementary of AUG. That's the start code, and then you have the second codon, and so forth. So as the first codon is read, it will move from the site and allow the second codon to be read. And as the second codon moves along, then you get the third codon. So the ribosome is slowly shifting, and it moves from the direction of 5' to 3'. Now, why is that important is because just like you have in English language, you read from left to right, which makes sense, and if you read from right to left, it makes no sense. So if the mRNA is read from the 3' end, it's going to code for a different sequence of amino acid, which will be a different modified protein. This process can be repeated as many times to ensure that you have enough amino acid in the sequence of forming a protein. Now, if you only had one ribosome at a time, then it will take forever. So in bacterial cells, what we have are polyribosomes that are like, if each of these little clusters that you see in black are each a ribosome. So when you have many ribosomes working together, you can have a cluster of ribosomes working, and you'll have many different proteins forming at the same time. So that's a polyribosome. Is it the same process in eukaryotes? Pretty much the same. Remember I talked about exon and introns. Eukaryotes have the same. It's called as mRNA processing. Besides that, in eukaryotes, two additional modifications actually take place. One is called as a 5' cap, and one is the 3' poly A tail. Now the reason for that is that the mRNA in a eukaryote will leave the nucleus and will reach the ribosomes, and in order to do that, it has to protect its nucleotide sequence. Otherwise, it would be chopped by the enzymes in the cytoplasm. So the 5' actually protects the mRNA from not being hydrolyzed by the hydrolytic enzymes in the cytoplasm, and the poly A tail ensures the same, so that it is protected I tell students about, like, think of the shoelace and how you have the caps on it. Think of the poly A tail and the cap, something like that. These are called as post-transcriptional modifications, because after transcription, after the mRNA is formed in the nucleus, and before it leaves the nucleus through the nuclear pore, these modifications are done. And thirdly, as I mentioned earlier, introns will be removed. Introns are regions that do not encode for any proteins. They have their own use, but they're not genetic material. They don't have any genetic code in there. So this concludes our second mini lecture on the genetics, and hopefully a lot of this was familiar from your other biology classes.